This is a production of Cornell University.
All right, so I'm going to talk about my research today, focused on understanding resistance to Phytophthora, root, and crown rot in squash and pumpkin. So I'm going to start by talking a little bit about the pathogen that causes this disease. It's a soil-borne oomycete pathogen called Phythophthora capsici, and it causes root, stem, and fruit rots on a variety of different plant species. So just a couple things I want to mention about the biology of this pathogen that have big implications in terms of disease management. The first of those is that when you have sexual reproduction happen with two different mating types of this pathogen, you get really hardy, thick-walled spores that can overwinter and survive dormant in the soil for many years, meaning that when this pathogen is introduced to a new field, it's almost impossible for a farmer to eradicate, and he or she needs to learn how to deal with it at that point. On the other hand, asexual reproduction produces much more short-lived spores, but they actually have flagella and can swim through the water. So when the soil is saturated, these zoospores are released, swim through the soil, find new plants to infect, and disease can escalate very rapidly given the right environmental conditions. So there are many plants that are susceptible to disease caused by Phytophthora capsici. The most economically important of those are solanaceous species, especially peppers, cucurbits, and some legumes as well. So I'm going to talk specifically about one of the host species of this pathogen, and that is Cucurbita pepo, which is one of the 3 species of squash and pumpkin that we grow in the United States. And this is actually the most economically valuable of those squash species. It includes modern classes like zucchini, other types of summer squash, acorn squash, and pumpkins. So Cucurbita pepo, compared to some other cucurbits, is particularly susceptible to root and crown rot caused by Phytophthora capsici. In other cucurbits like cucumbers, fruit rot is a bigger concern, but Cucurbita pepo is very sensitive in roots and stems. So this is what typical crown rot symptoms look like. Disease is first observed as a water-soaked lesion at the base of the stem. That'll then constrict the stem. The plant will die, and often you'll see sporulation on that dead tissue. So breeding for resistance to Phytophthora root and crown rot in squash has been very challenging, and there are no cultivars that are marketed as resistant to Phytophthora root and crown rot. And the biggest obstacle that we have is that we have yet to identify any sources of complete resistance in Cucurbita pepo. However, we do observe differences between different gene types in terms of how long it takes plants of those gene types to die. And that's what we're trying to take advantage of in our breeding program. So we've done a little bit of work trying to understand whether this can be characterized as resistance or tolerance. We've done some confocal microscopy using a GFP-tagged pathogen isolate, and we found that plants from genotypes that tend to survive longer also tend to have a less degree of pathogen colonization in their root tissue 3 days after inoculation. Similarly, we used quantitative PCR to estimate pathogen biomass in stem tissue 3 days after inoculation. Here I'm showing on the x-axis a visual rating of disease severity for 11 different genotypes. On the y-axis is the quantitative PCR-based measurement of pathogen biomass. And again, the genotypes that tend to have more severe disease symptoms, meaning that the plants die more quickly, also tend to have a greater degree of pathogen biomass in their tissue. So I think it's fair to describe this as resistance, where the plant is actually able to impede the growth of the pathogen in its tissue, rather than tolerance, where the pathogen survives, infects equally well, but the plant's able to deal with it somehow. I also want to mention quickly that you'll see really large standard deviation bars. Whether you're looking at visual ratings for disease severity or actually measuring the amount of pathogen in tissue, and that's an obstacle that we encounter with this pathosystem, where there's a great degree of plant-to-plant variation in terms of how it responds to inoculation. two plants of the same genotype, one might have 10,000 times more pathogen biomass in that plant at the same time point after inoculation. So it makes it very challenging to breed and particularly to select based on single plant phenotypes. Regardless, we've been making progress at Cornell breeding for increased resistance to Phythophthora crown root rot. We've used an accession called Austrian bush as a source of quantitative resistance. This is what's called a vegetable marrow type squash. Selection has been performed at the seedling stage in the greenhouse. And here on the right side of the slide, I'm showing some results from the 2 years of field trials we've done in the past 2 years. These are very condensed results. And in particular, I'm just showing 2 entries in these field trials. The reason for that is these 2 genotypes are the parents of the mapping population that I'll be talking about later. So this one entry is a susceptible zucchini cultivar called Dunja. We also have a Cornell breeding line. And you can see at 17 days after inoculation, some qualitative differences in terms of their response to inoculation. However, if you continue to observe those plants, you'll see I have that it takes, on average, about a week and a half to kill 50% of the Dunja plants in the field, whereas it takes a month to kill 50% of the plants of this particular breeding line. So we've made progress in selecting for increased resistance compared to susceptible commercial cultivars, but we still have a little bit of work to do in terms of getting a level of resistance that's economically relevant or acceptable for growers. In order to understand a little bit about the genetic architecture of this trait and develop markers that can be used in breeding, we performed QTL mapping. Like I said, the susceptible parent was the Dunja zucchini, and the resistant parent was actually an F4 selection that was the progenitor of that breeding line in the previous slide. So we elected to use a next generation sequencing bulk surrogate analysis approach. I'll talk a little bit about why we decided to do that, but first I'll just walk through the experimental design a little bit. So we evaluated 7,000 seedlings in the greenhouse, inoculated them with the pathogen, and selected the 15% of the population that were the most resistant individuals and the 15% most susceptible individuals. We also chose 15% random plants to serve as a control bulk. And we did this twice. So almost 14,000 plants, but we had 2 independently selected resistant and susceptible bulks of plants. So we took leaf punches from those plants, isolated and pooled their DNA, gave those DNA pools barcodes, and whole genome resequenced them on 2 lanes of the NextSeq 500 with paired end 150 base pair reads. So there are advantages and disadvantages to this type of QTL mapping approach compared to conventional QTL mapping. For our purposes, the main advantage is the phenotyping is relatively easy. Instead of assigning phenotypic scores to every individual in this population, we just had to identify the phenotypic extremes. So this enabled us to evaluate a larger population, which is important for this trait where we observe such low single plant heritabilities. So we use standard bioinformatic pipelines to trim the reads, align them to the reference genome, and call variants. We then filtered these SNPs to just include those that were fixed for opposite alleles in the parents, which we also sequenced. And we got rid of SNPs with extreme allele frequencies or extreme read depths. And I just want to mention that what we take from the sequence data from these pools is not a measure of a genotype on an individual, but the counts of the alleles that are sequenced. So if you're familiar with the VCF file, this is the part that we're interested in, the number of counts of the reference allele and the number of counts of the alternate allele for each pool and at each marker. So we achieved fairly high read depth, which we were pleased with, because with this type of experimental approach, the pools are composed of many individuals, and there's lots of error from sampling of reads during sequencing. So you need to have a high read depth in order to accurately estimate the allele frequencies at these markers. SNPs covered the whole genome fairly evenly, although there are some regions better represented than others. So this is kind of busy, but I'll try to focus on some of the regions in a little bit. I'm just going to introduce what I'm showing here. And this is the frequency of the allele derived from the resistant parent in each of the 5 DNA pools, 2 susceptible pools, 2 resistant pools, and 1 random pool. In any region that's not in LD with a polymorphism that affects the trait, we expect allele frequencies in these 5 pools to be the same, taking into account some noise from sampling individuals and from sequencing error. So that's what we see, for example, on chromosome 3. The frequencies in these 5 pools they fluctuate across the chromosome, but they don't deviate from one another. On chromosome 4, however, if you look at the end of the chromosome, you'll see that the 2 resistant pools in green are enriched for the allele that's inherited from the resistant parent, and the 2 susceptible pools are depleted for that allele, and the random pool has intermediate allele frequencies, which is a pretty convincing signature of some polymorphism in that region affecting the trait that we selected upon. We see something similar at the beginning of chromosome 5. On chromosome 8, we see a similar pattern in that the resistant and susceptible pools have different allele frequencies, but we observe all the pools to deviate in the same direction from 0.5. And this could be because of segregation distortion or some artifact from mapping combined with a polymorphism that affects differences, the frequency in each pool. So chromosome 16, at the end of chromosome 16, we observe similar signature. However, here you'll notice that the resistant pools, I should say, are actually enriched for the allele that's derived from the susceptible parent, suggesting that there are good alleles from both parents of the mapping population. So many ways have been published to perform hypothesis testing with this type of experimental design and data set. This is one method which I'm showing here called multipool, which takes into account recombination rates and all the markers simultaneously on a chromosome to estimate LOD scores in 100 base pair intervals. And as you'll see, whether you use the data just from rep 1 or rep 2, we have large peaks in those regions I just mentioned, chromosomes 4, 5, 8, and 16. You get higher peaks when you combine data from both of the reps because you have a larger sample size and greater read depth. There are also other regions where we observe minor peaks consistently between both of the reps, suggesting that there are probably other minor QTL involved, but we don't have the statistical power to confidently declare QTL there. So what did we learn from this experiment? Well, we learned that resistance appears polygenic. There are desirable alleles from both parents. And with this type of experimental design, we're not able to actually estimate QTL effect sizes, but we do observe that even in the regions with the strongest signal, the absolute magnitude in terms of the allele frequency differences between the pools was fairly small, around 10 to 15%. So this could be because of poor accuracy in visual selection, again, because of all the plant-to-plant variation, or could be because the actual effect sizes of these QTL are fairly small. So in order to evaluate how effective marker-assisted selection can be for Phythophthora root and crown rot resistance in squash, we're currently developing an F2-3 population. We'll be able to use the genotypes of the F2 parents and the phenotypes of their F3 progeny to compare the predictive performance of markers in QTL regions to whole genome markers using whole genome prediction models. So with that, I'd like to thank all the members of the SMART and GORE labs, my committee members, Mike Gore, Chris Smart, Michael Mazurek, and Philip Messer, Kyle LaPlante from the Mazurek lab, who's worked on a lot of the breeding for resistance in squash. Summer scholars, Ella Reeves and Jessica Helvig, who did a lot of the qPCR and microscopy experiments, and funding from New York State Department of Agricultural Markets and the USDA. Thank you. Yes.
When you sequenced your F2 books, did you sequence each of the books with a different tag? Not each book, but with each individual within the book in a different tag?
No, there are only 2 DNA samples. So each sample represented DNA pooled from all those individuals within that breadth. Yes, Dan.
Did you tissue sample your susceptible lines on the way out, or did you tissue sample everything and then decide what was resistant and susceptible?
We just sampled those individuals that were selected. So we waited a couple days after inoculation, found those plants that first started to wilt first, and took tissue punches from those samples. And then we waited another 5 to 7 days for the plants that held out the longest and selected those as the resistant bulk. Yeah.
This comes up every time you give a talk on this pathogen, but since it's it has almost obligate like sexual reproduction, could you flood a field with like sterilized spores of one mating type to like just purge it, like people would do with mosquitoes? Like if ever dudes, it's just a gene drive. But
That's an interesting idea. I haven't really thought about that. It might be hard to convince a farmer to flood their field with pathogen.
Mutation. I mean, like people did, again, they released like tons and tons of male sterile mosquitoes.
Right. Yeah, that's an interesting idea. It might be hard to, maybe in like a newly infected field, but the oospores, the sexual spores, they vary in how long they take to germinate. And we have evidence of oospores germinating 7 years later and starting infecting plants. So if you already have that bank of spores in the soil, it might be kind of hard to overpower that, but maybe with a newly infected field. All right, thank you.
This has been a production of Cornell University, on the web at cornell.edu.